David Sinclair is up next, and he's getting set up here. David is professor of genetics at Harvard Medical School and co-director of the Paul F. Glenn Center for Biological Mechanisms of Aging. He's the co-founder of biotech companies including Certis and Life Biosciences, head of the Sinclair lab at Harvard, and the go-to guy for aging. Thank you very much. Or not aging.
Not aging, yeah. Thanks, Joe. Thanks. How do I say it? Sapiens? Sapiens? Actually, Joe, you might be more right than you think. We're able to speed up aging, we think, and slow it down, which I'll tell you about today. But it's great to be here. Typically, I talk to biologists, and so I have to focus very narrowly on biology and how they think. I think this will be great. I'll be able to open up a little bit and tell you my broader mind. I also come up with ideas in my sleep, so this is great. Problem is when you have kids, you get less sleep, so it doesn't work so well. Yeah, that's just the disclosure stuff. So I want to start by saying we are in line to have an amazing future. I've seen things in the last few years that I never thought I'd see in my lifetime, including in the aging field, which we work on. And of course, there's a lot of speculations on what human evolution will be, but it's not going to be like that. It's not, at least not in the very near future. What we're going to see within our lifetimes and in the coming 10 or so generations is not an evolution that happens naturally. We are now in charge of our own evolution. That's very clear. There's very little evolution happening across the planet. There's too many of us. And natural selection doesn't have much force in large groups. So it's really in our hands. That's what's important to know. And I want to tell you a little bit about the world that I inhabit with the design of actually controlling our own evolution. So this is the inside of a plane. I think we've all flown, most of us. And I was on a flight recently coming back from Florida. And I was talking to somebody about what I do. And they said, what do you do? And I said, we work on aging. And they said, why would you work on aging? And I said, well, it's a really big problem. And most diseases that kill people in the world are caused by aging. If you smoke, it increases your chances of getting cancer 5-fold. If you age, it goes up 1,000-fold, which is more important. And he still looked at me and he said, but it just sounds so unnatural to work on aging. And I said, what are you talking about? We're drinking alcohol. We're texting our wives. We're at 30,000 feet in comfortable atmosphere. What about any of this is natural? And I would put it to you, what in this room is natural, except that we're breathing oxygen? That's about it. So let's just throw away what's natural and let's go with what's best for us as a species and best for the planet and our cohabitants on the planet. So there's a next-gen human coming, and there's plenty of ways to improve ourselves and the world we live in. Some of the technology I'll tell you about today isn't just applicable to our bodies, but to the animal world, to the plant world as well, and certainly bacteria. Later we can talk about, perhaps in the question time, about what impact this will actually have on society, but I don't have time to talk about that just yet. And I'm going to touch on about 5 of these various ways I think that we're headed for human improvement. A lot of this is going to happen in our lifetime, and that's the amazing thing. Sometimes I talk to the media, and reporters will ask me, is this real? Is this real science? And I get frustrated because we've been, as a field, publishing in the top biology journals for the last 25, 30 years. And it's as if the world hasn't yet heard about it yet. But they're very quickly waking up to the fact that this is going to happen, whether they like it or not. So AI, I feel like I'm in a room of people who know way more than I do, but I'm certainly interested in AI. And what I am mostly interested in is what are humans going to do with it from a medical standpoint, but also to be able to utilize brains that are better than ours. This is Henry Markram. He's at EFPL in Geneva. Probably some of you know him. I was just over there. This is from a few weeks ago. I wanted to see what I thought was cutting edge. And I really was impressed. What he's doing is modeling the way neurons interact. And he's built certain parts of the mouse brain already. And he gets emergent properties of what appears to be thinking beyond your regular machine learning / neural net work. And so I'm certainly on board with the with the panel, and I disagree with Elon that this is not going to happen. I think it's going to happen way quicker than we thought, and many of us think. A lot of what Henry's doing can fit on an iPhone. All right, so what about the future? Well, part of our future has to be off this planet, and this is what Mars life will be like. The other problem, and I think Elon fails to at least think about it or talk about it enough, is what's going to happen to our bodies when we get there? And that, it's a horrible place out there. We're very lucky to live on a planet with a magnetosphere. And one of the big problems, and I work with NASA, big problem is that our DNA and our control program of DNA, the epigenome, is going to be toast. It's about a 900-day journey back and forth, and that's enough time that you're going to come back with dementia, premature aging, probably some Alzheimer's, just to say the minimum. So one of the things that we've been working on is, as a way of using the technology that we develop in my lab, is to use that to actually help people get to Mars and come back without dying. And so we submitted a proposal to NASA, and one of the things that NASA was looking for was a way to protect astronauts in near space and deep space, and certainly on Mars. And one of their big problems is, how do you shield a human being? And some of the proposals were put a meter of water around the spacecraft, or about half a foot of lead, which is fairly heavy. One of the ideas that we had was, why don't we protect the cells, protect the genome, and shield that? And we have ways of doing that. There are actually some new innovative ways to be able to prevent cells from getting damaged, and as well as repair more efficiently. So I'll tell you a little bit about that. The molecule that is all the rage in my lab is this one. This is called NMN. A version of this is in clinical trials over at Brigham and Women's Hospital. Been feeding this to humans for about 18 months now. All has gone well so far. And this is the molecule we had a science paper a few years ago showing that this actual molecule, at least in mice, protects those mice from DNA damage and restores the DNA repair capacity of an old mouse to a young one again. And that's an important theme. Often we think that aging is a one-way street, and what we've learned is that there are aspects of aging that are remarkably reversible. The mice that we published on were treated just for a week, and that was enough to reset that part of the program. I don't know if you can see black mice on a black background, but there's a mouse on the top left and a mouse on the bottom right. The one on the bottom right has stopped running. So either it's lost its energy or it's just a lot more intelligent. We're not sure. But we assume that it's because it's tired. It certainly has more lactic acid in its bloodstream. And what we found, and we published, this is now a paper from exactly a year ago, was that this same molecule can intervene in the loss of endothelial function of the capillaries in your body. And actually, a very poorly studied aspect of biology is the loss of capillaries and blood flow as we get older. And that underlies a lot of chronic disease, actually. But the reason that we haven't done much about it is we thought that that was also a one-way street. Even exercise, after a while, doesn't affect the body's ability to grow new blood vessels. So we thought that was just a natural part of aging. We tend to accept what we cannot treat. But these mice, they grew new blood vessels in their muscle, they grew blood vessels, we think, in their brain, and there was no evidence of any downside yet. We've tested cancer, it didn't seem to have any negative effect yet. But this is pretty interesting. A lot of sports associations have reached out. Oh, I should mention, these are old mice. I'm not sure you read that, but we can make an old mouse run just like a young mouse again after just a few weeks of treatment. Which is really quite a remarkable thing if you think about it. The other thing that NMN, and I'll tell you how NMN works in a minute. The other thing NMN actually did was we study fertility in the lab. And one of the things that I'm convinced is that the ovary, being the first to fail in mammals, can be protected and possibly even reversed in its aging process. And there are a lot of women, I'm sure, in the audience who realize that having a career interferes with having a family, and it's sometimes too late. And so what we're working on are ways to improve fertility. And these are mouse eggs. I don't know if you can see that there's a red cytoplasm, but what these blue things are the chromosomes lined up ready for fertilization. And unless your chromosomes are nicely lined up with the spindles in green to pull them apart, you're not going to have a healthy egg or an embryo that survives. when you get old, if you're a mouse and a human, this is a 16-month-old egg, and you can see its chromosomes are never going to produce an offspring. And that's one of the problems for women when they become older. Their eggs are either gone or their chromosomes are not nicely lined up. But what was quite amazing was just after, again, 4 weeks of treatment of females, these mice produced healthy eggs, and we could have much more likely to have offspring. And one of the things that's quite confusing, and we're just trying to publish this work now, is that we can treat mice that have lost their fertility and they regain their ability to have offspring. And those offspring appear to be quite healthy over many generations. So if I haven't convinced you yet that aging research is relevant to other things, then you may as well leave now. Or I can leave, one of the two. So how does NMN work? There's a lot of stuff in the literature if you want to read about it. Basically, we found a set of enzymes. When I worked down the hall here at MIT 20 years ago, in the Goin lab, we discovered that there are a set of enzymes that control gene expression and cell defenses. We call these the sirtuins. And we have 7 of those genes in our body. And those enzymes that control genome stability and the epigenome need NAD. NAD, you'll remember from biology, hopefully, is a molecule that's required for many enzymatic reactions. Without it, we're dead in about 30 seconds. This molecule goes up and down during the day. When you wake up, you get a boost. When you want to go to sleep, you go down again. NMN is the immediate precursor that our bodies use to make NAD. So when we're giving these mice NMN and the humans that are in clinical trials, their NAD levels go back up. And what I haven't told you is by the time you're my age, which is 50, we have about we 50-year-olds have about half the levels of NAD that we had when we were 20. So what we're hoping to do is just by raising NAD levels, we can get the body to fight against disease and debilitation or deterioration a lot better. And it seems to be working, at least in mice so far. And the older you are, the better it seems to work. And those mice we have in a study ongoing, but it looks like they're certainly healthier and possibly longer lived. All right. So what about gene editing? This is an area that is certainly controversial, but no biologist talking about the future of humanity would be worth their weight in salt if they didn't raise it. And I'm sure that George is going to talk about it next, having co-invented some of the tools. This is just a slide to remind me to say that we're looking at the effect of genes and the ability to deliver genes in mammals and now in humans to be able to correct but also augment what we were born with. Okay? And hopefully I won't forget to say, so I'll say it now. We are collaborating with George Church, who you'll hear in a minute, on this. So instead of altering the germline, which I think you all know is possible but frowned upon, this is another way of doing it. You can use viruses to deliver genes, or you can just introduce RNA, which is not going to be a permanent effect. And more we're learning how to do that as well and not use viruses. But for now, the FDA allows us to use viruses. Typically, they're used either in the bloodstream or in the eye to correct genetic diseases. And there's a flood of technologies being developed in this area, and many companies are working on this. So this is an adeno-associated virus that we use in my lab to introduce viruses into an entire old mouse and see what happens. But which genes would you want to use? So let me tell you a bit about that. So some of the genes that we work on in the lab come from other species. We think that we're not perfect, and some of the best genes are from other animals. And so this is a tardigrade that will survive in outer space for a little bit in the vacuum of space. So we've got the genome of that. We've cloned genes out of that organism, and we've found genes that seem to protect mammalian cells dramatically against DNA damage. Venkat V, I won't pronounce his last name because it's way too long and difficult, but Venkat works at JPL, so we're collaborating with JPL on their collection of thousands of extremophiles, fungi and bacteria that survive in Chernobyl, out in space, deep in the earth. And Kyle Landry is our expeditionary scientist who goes and finds these extra bugs. He's about to go down a few miles into the Earth's crust in the next few weeks. And so together, Kyle and Venkat in his lab are working on finding genes that can augment plants, animals, and humans. One of the bugs we're working on, and this in disclosure and transparency, is being done at a company out in Worcester, Massachusetts, called Liberty Biosecurity. And this is a company that, or this is a bug, I should say, that is able to survive hundreds up to 1,000-fold more UV than a typical life form. And so there's a bacillus pumilo, and it's found all over the spacecraft that come back from space. And the Russians have found it. And you can see that's a Russian, I probably can't see, but that's Russian language up there. And this is actually, these are called coupons. They put the bacteria on these metal strips and they expose them to outer space and look at how long they survive. And Venkat's team has published that these can survive out in space for over a year. So they got something very special about them, and we're cloning genes out of that organism as well. But humans have pretty good genes too. We don't all have the best varieties, but some people do. And so at least 25% of human longevity is genetic. And these families that tend to live a long time, these centenarian families we call them, they have genes and gene variants that we're cloning out. One of these sirtuin genes that I mentioned is actually associated with longevity, number SIRT6 it's called. And so we're looking at making mice. Actually, if you put SIRT6 into mice, they live longer, so that's a good news from an ex-postdoc from my lab, Chaim Cohen. And so with all that knowledge, we're hoping to be able to fix what we were not given by evolution. And in the final few minutes, I want to talk about what I think probably drives a large part of our aging process. And so we used to think back in the 1950s, 60s, 70s, guys, people like Szilard, Medawar, they proposed the DNA damage hypothesis of aging. And after that, we had others that proposed that free radicals were the main damaging agent. Anyone here still think free radicals is the main cause of aging? Well, it's still a billion-dollar industry, but we don't think in the aging field that that's right. Now, there is a role for them, but some of it's actually helpful. What we think in my lab is that aging is certainly a loss of information, but it's not the genetic information, it's the epigenetic information, the ability to read the right genes at the right time. And we actually think in my lab of the body as having two main types of information. The digital code, which is long-lasting, you can get it from Neanderthals. It's actually, DNA is a very good way of storing information. It's base 4 rather than binary, ATCG. That doesn't appear to be the main cause of aging. Old people have, or even middle-aged animals have enough information to be able to clone and make new animals that actually live normal lives. So just that observation alone, to me, says aging cannot be largely driven by the loss of digital genetic information. So what's the other information? It's this epigenetic information, which includes how the DNA is wrapped, how those larger things fold into structures. The problem with epigenetic information is that it's analog. And anyone who's, probably most of you haven't owned a record player, but anyone who had a record player, or certainly a cassette tape, knows that it's not very good at storing information over time. Even if you copy it, you certainly lose it, but over time, just cosmic rays will obliterate the information. So let me give you evidence that we've been working on that epigenetic change and the loss of epigenetic information is a driver of aging. So I'm at MIT, so I thought that I'd show you a slide of one of my idols, Claude Shannon, who was here in the 1950s. His paper from the Bell Labs in 1948 is one of my favorite. Probably many of you here have heard about it. This couple of papers in 1948 set the stage for the entire internet. It was the first mathematical theory of, well, one of the first mathematical theories of communication. And what he's showing here is that you've got a transmitter and a receiver, and there's always noise that comes in. And he knew that noise was a major problem. He'd lived through World War II. He was part of the people that part of encryption and decoding, and he knew that the loss of information could cost lives. And so he worked on this and came up with mathematics that allowed information to be passed along quite effectively. And he was a big proponent of digital information. So this is now a cell, and we see that some genes are off. We call that heterochromatin. Some genes are on, that's euchromatin. And over time, we as a field see that these cells start to lose their gene expression patterns. So what you get during development, when you're 15, 20, that pattern of gene expression starts to become noisy, and cells lose their identity. So that's really not a big argument. The question is, why does that happen? Why do you get epigenetic noise? Why do we succumb to second law of thermodynamics, even though we're eating food and using energy? For those of you who have never seen one of these, this is called a compact disc. We used to store songs on it, I think. Anyway, so what we think is going on in aging is the equivalent of the digital information still there, but the scratches are not allowing the right genes, the right information to be read at the right time. So what do you do about that? Well, first of all, you can test, do these scratches cause aging? So we have a mouse where we can introduce epigenetic noise. We do that, I can talk about it a bit more later, but essentially we cut the genome and force the DNA to be reorganized. And so in the repair process, eventually cells lose their organization of the genome. We don't find mutations, so these mice that I'm showing you are actually essentially genetically identical. They're twins, black 6 mice, and they're the same age. They're 16 months old. And at 10 months after we've created this epigenetic noise, we get what appears to be a very good close approximation of aging itself, both at the physiological and the molecular level. And there's actually a DNA methylation clock. Has anyone heard of the DNA clock? Probably. You can have a blood test now and measure the DNA methylation pattern on your DNA and predict how old you are, but no one knows what drives that clock. These mice get older based on the methylation clock. So we think that epigenetic noise is a driver of that process, and a large cause of that noise are breaks to the chromosome that need to be repaired in the reorganization of the epigenome. So those ice mice, there's the clock, shows that it's that these mice are getting older. We've measured muscle and a variety of other tissues. But how do you get those mice to go back to normal? That would be the real holy grail, or swinging for the fences. So we've been working on that for a while. Essentially, this would be like polishing your compact disc, so your cells can read the original information the way they did when they were young. Again, we refer back to Claude Shannon. This is from his other paper in 1948, and what he showed brilliantly was that the best way to preserve information if you lose it at the receiver is to go back to an observer who holds the original information, send a signal here to retrieve the correction data, and then you end up with a restoration of the signal. And that's exactly, actually, the way the internet works, is Tim Berners-Lee. And you can see that there are all these correction protocols. Your emails don't often come through unscathed. You have to go back and get the packets. I think the same is going on with aging, in that there's a transmitter and a receiver. Transmitter and the source are your cells when you're young, and the receiver is your body when it's older, and you've had noise introduced in the meantime. But imagine if there was this observer in our cells that could say, hey, when you're triggered, bring back the correction data and correct your epigenome. So one of the ways we tried was to reprogram cells. Now, I think many of you know there was a Nobel Prize awarded in 2012 for cellular reprogramming to make adult cells back into a pluripotent stem cell. Now, nobody for a long time thought it was sensible to put these genes into a living animal. You'd end up with the world's biggest tumor, about the size of a mouse or a human. But we did it anyway. And so Juan Carlos Belmontes at the Salk Institute deserves credit for being the first person to do this in a mouse. He created a transgenic mouse and put the 4 what are called Yamanaka factors in and triggered them to be on. What he ended up 2 days later was a dead mouse. Okay, so that's a bit of a problem. But what he found was if you let the mice recover after those 2 days and then hit them again a week later, they actually survived and they did better. And you could actually have a longer-lived mouse in the case of a premature aging mouse model. But the problem still was that it's very toxic. And so what we decided to do with George was to remove this gene here, c-Myc, which is an oncogene. Doesn't take a genius. And we put these other 3 genes in. Now, this was not easy to do because we wanted to deliver them with one of those adenoviruses. And adenoviruses don't typically fit more than 1, perhaps 2 genes. But my student, Wencheng Liu, who's quite a clever student, he was able to whittle down these genes, whittle down the virus packaging, or the plasmid, and actually fit all 3 in. So now we could ask the question, if you introduce 3 of these 4 Yamanaka factors into a mouse, what would happen? Do they die? Did they become a tumor? Or do you get rejuvenation? Or nothing, I guess, could have happened. So we put them into mice. They didn't die. They're now about 9 months. It's 9 months later. They seem to be quite healthy, no tumors. and we're waiting to see how long they live. But in the meantime, we've been testing it in parts of the body that typically don't regenerate. The part of the body that typically doesn't regenerate after you are about a few days old are your nerves. Typically, if you crush your optic nerve or you break your spine, you're not going to walk again or see again. And people have been working for a while and trying to figure out if you have a crushed optic nerve, this being a mouse's optic nerve, what happens. So what you can do is you can put in a dye, you can give a virus, you can crush the optic nerve and have a look, either looking at the retina or at the side view of the optic nerve, do you get any regrowth? And this was the first data we got that showed that if you reprogram these cells actually after the crush, you could preserve them, so it's a lot brighter there, so a lot of the nerves survived, and they started to regrow, so the brain is down here somewhere. This was after, this was 1 month. If we leave this for 3 months, this fills in and is really bright, and actually these nerves make it all the way back to the brain. Now, we don't know if those mice can see, but we've now tested this same reprogramming on old mice that have lost their vision, as well as glaucoma, which is the pressure in the eye that damages the retina. And in both those cases, we've restored vision, which is something that you wouldn't have expected to be that simple. What we think is going on is that these nerves, at least in the old mouse, are dysfunctional. They're turning on the wrong genes at the wrong time. That's aging. And we can reestablish the original youthful gene expression program by telling the cell, go get the stored information and make me young again. And we know that part of this is to do with the DNA clock, the methylation on the DNA, because if we block the enzymes that remove those methyl groups, this doesn't work anymore. Okay? But what actually tells a cell remove that methyl group and not these others, that we don't know yet. But that's a major challenge. And there's one chain. So we're going forward, we want to be able to treat glaucoma patients initially, so probably in 18 months, 2 years, if we're lucky, we'll go into our first clinical trial. But we only chose the eye, remember, because we thought it would be interesting, not because it would work there. Theoretically, imagine a time when you're young, here's a young Bill Murray, you could have an injection of OSK or some genes like it. These are all inducible, I didn't tell you that, but you can just give the mouse doxycycline and on come the genes, take it away, turns off again, very tight. So why not have this injected when you're young? Now you're a transgenic human. And when you get into trouble, you break your spine, you get sick, you get old, you take a course of antibiotics, doxycycline, and perhaps Bill Murray would go back to his former self. Who knows? But this is just one of the ways I wanted to stimulate your minds, get the discussion going of what's possible for the future. And then finally, I also agreed about the ability to get off the planet. It's something that we should be working on, and thank heavens for Elon. The problem is, unless we're able to send packets of information, say in a SanDisk little packet, the problem is that we weigh too much. And unless we want to freeze dry ourselves, we can have about 30,000 years to get to the nearest habitable planet, by my estimations anyway. Anyway, it's going to be a lot. It's going to be thousands of years at least. So one idea is that we can evolve ourselves to be able to make that flight or send eggs. That would also work. But if we want to do it, we would have to engineer ourselves to be long-lived, robust, survive radiation. But also what's important to remember is we can send information much quicker than we can send ourselves. So innovations that happen on planet Earth can be sent to the spacecraft, and hopefully on board they have the ability to use that technology and become much more advanced as they head off to the planet. But eventually they'll need, when they get there, they'll probably be more like a new species, having re-engineered themselves. And the question is, would you want to reverse that or not? I'll leave that up to you. So finally, there's a lot of people I want to thank in these slides. I won't go through them, but there's George, I mentioned him, and plenty of others have helped along the way. And thanks for inviting me to this. Appreciate it.
Thank you, David. It was wonderful. There's one question that came repeatedly from the audience, and as an aging member of the audience, of course, I would ask it too. Is NMN safe for humans? And if so, can people get it? Or when will people get it? Is it the new resveratrol? If so, when will it come?
All right. I get hundreds of emails a day. Yeah.
That's the obvious question.
All right, first I'm going to go with the disclaimer, because I'm a professor at Harvard Medical School. I don't recommend anything. I don't recommend any products. I certainly don't endorse them, even though you can find my name all over the internet. And I do send out cease and desist letters a lot. OK, with that said, NMN is available on the internet. I haven't tested those products, so I don't know what is in them. There are people selling a similar molecule to NMN called NR, nicotinamide riboside. They both raise NAD levels in humans and mice, and they both have had some health benefits. We don't know yet. I'm hoping by the end of this year I'll be able to say more with the clinical trials going into phase 2. Is it safe? In mice, it's very safe. They live longer. There can't be any better evidence than that. There's always the worry that if you have an existing tumor, it may grow more blood vessels, so I'd be careful. That's one warning, but I haven't seen any evidence that cancer is provoked in mice, at least. But we'll see. I'm on record as having taken NMN. I'm not dead yet.
Okay, well, it's wonderful to hear, and we'll stay tuned. There's one other question, and it's something you alluded to a bit. Well, there are a bunch of questions, but this one was intriguing. Is the person who's going to live to be 150 years old already maybe here in the audience or born? And then my corollary to that, what's the upper limit? Will Ray Kurzweil make it?
Oh,
right.
I hope Ray makes it.
Yeah, we all root for him, right?
Yeah.
He's the poster boy for this.
He is. Well, I think it's feasible that somebody could make it to 150. Remember, people born today will, even if we don't succeed, will still see the year, 2120. That's a long way in the future. A lot is going to happen in those years. What were we doing 100 years ago? We were learning how to fly, and we didn't even have antibiotics. So remember that it's not today's technology that future or people living today will have. They'll have things that we can't even dream of. But I get criticized for saying that somebody could live to 150. But I also am of the belief that you should never say never. I think it's feasible. What I think is really realistic is that more and more people are going to make it to 90 and 100 in a healthy way. Yeah. We tend to think, we look at people who are 90 and 100 and think, why would I ever want to reach that age? But what we should remember is getting to that point, first of all, it means that you can be healthy 60, 70, 80, 90, playing tennis, hanging out with your great-grandkids. That's important. So we're extending out the health span, not just lifespan. But perhaps those 90, 100-year-olds who make it there are going to be living much healthier lives. And what's interesting is the mice that make it out there into that later life, what we find is that they die more quickly. So we're rectangularizing the mortality curve, which is something that I think any sensible person would think is a good thing.
There was that study recently that said that humans, it was looking at statistics, there's a sharp cutoff in human longevity at about 120 years, something like that. I think it was a French study. Is there any gist to that, or?
Yeah, so the longest lived person, and even that's debated, is 122. Yeah. There appears to be a barrier, but none of these people had done anything except perhaps drink red wine and have a sense of humor. We have better technology than that now, so who knows what's possible.
Maybe the humor gets better. On that note, thank you very much. I think we have the break now, and